Welcome to the channel Synaptic Signals. Today we are going to learn about the chapter Heredity. Now, this is a very important chapter for your exams. For the SEE, Heredity carries 16 marks, which makes it one of the highest weighted chapters in science. By the end of this lesson, you will clearly understand the key concepts of heredity and be better prepared to tackle exam questions with confidence. So stay with me till the end because we will cover all the important topics step by step. Alright, let's begin. The first thing we need to understand before diving into heredity it is chromosomes. So let's dive into the topic chromosomes. What are chromosomes? Chromosome is a thread-like structure which is made up of DNA and proteins, so mainly histones. So if I ask you like where are these chromosomes found? So chromosomes are found in the nucleus of the cells. So what does it do? It carries genetic information that determines the traits of an organisms. So it becomes visible only during cell divisions when chromatin threads condenses. So as you all can see in the screens, the purple line denotes the chromatid and the middle line denotes the centromere, orange color one. Now this structure clearly shows the X-shaped structure of the chromosomes, which have two identical chromatids joined by centromeres and tightly packed DNA which carries out genes, the actual units of heredity. Now it has two arms, which is like they are divided into two parts, the short arm, which is the P arm, and the longer arm, which is the Q arm, which are protecting our genetic informations. Now, if you look carefully at this diagram, you will also notice that there is a long word over there that is synaptonymal complex. So it is a special structure that forms during meiosis when chromosomes pairs up with their homologous partners. It plays an important role in exchanging genetic materials, which we call crossing over. This is why we don't look exactly as our parents or siblings. It increases genetic variations. So summarizing, chromosome is made up of chromatids joined by centromere with equal arms. During meiosis, synaptic complex helps in exchanging of genes. Now in this slide, you can see the different types of chromosomes on the basis of the positions of centromere. So the first one is metacentric, having appearing like both the arms are equal and X shape, and also submetacentric chromosome, which has one arm is shorter and other is the longer one. When the centromere is very close to the one end, it forms acrocentric chromosomes, which is the third picture, giving one very short and one very long arm. So finally, the last one is the telocentric, where the centromere is right at the tip. So we call it a telocentric chromosome. As it appears, it has only one arm. So these structural differences are very important in genetics and helps in identifying chromosomes using karyotyping. So this was a short video explaining to you about structure and types of chromosomes. Thank you for your patience, for listening and watching my video.